Hello everyone. Today's topic is production of human insulin by rDNA technology. First of all, what is insulin? Insulin is a chemical substance or a proteinaceous substance, or you can say these are hormones which is produced in our body by its own. From where it produce? It produce from the pancreas. In the pancreas by the islets of Langerhans. And what is the function? What is the function of insulin? That is regulates or control the amounts of glucose in the blood. That means it's control the blood glucose level or blood glucose concentrations. See, when we take some food, after simplification, it converts into some carbohydrates, right? This carbohydrate is uptake by the cell from the blood, right? Insulins acts like a MAKAUT for entering those carbohydrates into the cell. That's why we need these insulins in our body. Otherwise, the concentrations of the carbohydrates or glucose increase in our blood, and it creates some disease like diabetes, hypertension. That's why we need a proper amount of insulin in our body, right? Normally our body can create some proteins by its own. In this aspect, when our body will not produce these proteins which we require, is that we need to uptake those proteins from outside. Like when our body is unable to create sufficient amount of insulins, we can uptake those insulin from outside. Let's see how insulin looks like. Basically, when insulin produced by pancreas, it's produce the insulin as a proinsulin. Proinsulins mean it is the inactive forms of insulin. So what we need to do? We need to convert those proinsulins into the insulin, right? We can convert the proinsulin into insulins by the post-translational modifications, like translational modification. By this post-translational modifications, we can convert the proinsulins into the insulin forms. That is, from the inactive forms to active forms. And what is the difference, major difference in between the proinsulin and insulin? That is, proinsulins contains 3 peptide chains, that is peptides A, which is contains almost 21 amino acids, and peptide B, which is contains with 30 amino acid, and the peptide C chains, which is act like a connectors in between the peptide A and peptide B chains. In case of insulin, that is active forms of insulins, it's contains with the peptides A chains and peptide B chains. In between the peptide A and peptide B chains, there is the presence of disulfides bonds. And the next, Humulin is the first human insulins for diabetes treatments created using the recombinant DNA technology. In October 1982, it is approved by USA Food and Drug Control Administration, that is US FDA. And this insulin is created by the company Eli Lilly. Let's see the process. Let's see the process or technology behind the insulin preparations. See, for production of insulin in lab, we need a vectors. Here, for vectors, we can take a bacterial cells which is contains with 2 types of DNA, that is external DNA, it is called plasmids, and the central DNA, that is circulate DNA or bacterial DNA. Plasmid as a vectors, and also we need a human cell from where insulin genes we need to extract. But previously, That means the insulin gene was isolated from various animals like sheep, cow, but nowadays it is isolated from the human cells or human DNA that is responsible for insulin productions. Next, through the help of restriction endonuclease, we can cut those gene or required gene from the human DNA, and for and with the same genes we can cut the plasmid, that is vectors. Both DNA can cut as a sticky end. After that, we insert the insulin genes into the vectors, and through the help of DNA ligase sells the human genes and plasmids, and it forms the recombinant DNA. This recombinant DNA enters into the host cells, and host cells clone the daughter cells, which are cultured in fermenters. After culturing, so we extract the desired genes or desired insulins for further use. Basically, what happened in this technology, that we need a human cells which contains the gene insulin or insulin genes, and bacterial cells, that is E. coli, that is that contains the plasmids and the bacterial DNA, from which we can take the plasmid as a vectors, and from the human cells we can isolated the desired genes, that is insulin containing genes. After getting those kind of DNA strand with the help of restriction endonuclease, uh, insulin DNA or gene of interest included into the plasmids or vectors. With the help of DNA ligase, we can create the recombinant DNA. This recombinant DNA entered into the host cells. This host cells can cultures into the fermenters, and those host cells create some daughter cells which contains with the rDNA, that is recombinant DNA. After creating these daughter cells, we can extract those desires or gene of interest and collect for use. So that's all. Hope this video will help you. Thank you for watching.